We'll wait for your confirmation of thrust before releasing the ROV.
Copy that. Pilot has control.
Pull the pin. Stand by for line, pilot. Okay, line's clear, pilot. If you can make your way to center line and push ahead, please.
Copy that.
That's good heading there, pilot. Looking good, pilot. Let me know when you have good visual in the starboard robo.
Yep, good visual, straightening out.
Great.
Auto headings in, off stick otherwise this time.
Copy all, pilot. We're going to come down on our forward speed a little. Otherwise, looks good.
Copy that.
Okay, deck. Our streaming looks good. Let's launch Sirius. And pilot, if you could provide some slack as necessary
Copy
when
that.
the Sirius goes over the transom, please.
Understood.
That's looking good, pretty good, pilot. Let's maintain that.
Copy that. Maintaining. Pilot's off stick.
Copy, pilot off stick.
Turns have been reset.
Roger. Winch 0 the counter, please.
Counter 0.
Deck disable A-frame, please. Thank you. Okay, pilot, are you ready to continue with the dive?
Yes, sir.
Okay, let's go down to 50 meters and hold at 15 meters a minute. Winch will do the same.
Copy that.
One 0 meters, wire out.
D2 just past 1.5.
1-5 I. Two 0 meters, wire out.
D2 27 meters.
27 I. Three 0 meters, wire out.
D2 36 meters.
Four 0 meters, wire out.
D2 4 or 5 meters.
Dive suit nav, we have tracking on D2, still waiting on Sirius.
Understood, thanks, Nav.
D2 stopping just below 50 meters.
Good copy, D2 at 50. Winch, I'll stop 50 meters. Winch, dive super.
Good, yes.
We pass control to the control room, please.
Control and mass control.
Control confirms control of the winch.
Copy, co-pilot. Nav, I think before we pass ship control, I'd like to get tracking on Sirius. So maybe we'll just keep it as is while you guys troubleshoot.
Understood, dive suit. Dive suit nav, just update. We're running troubleshooting right now, trying to wake up Sirius one more time.
Okay. Sounds good. We'll maintain until we get that.
Roger that. Dive suit nav, we have good tracking both vehicles.
Copy that, Nav. Pilot, Dive Super.
Dive super go.
Hey, you guys are free to continue with the dive. I'll work with Bridge to take control, but I'll keep comms. You guys are free to dive.
Understood. Thank you.
Okeanos has joined the call. We're just waiting for the vehicles to continue the descent and for the scientists to get in position. those on the ship and the shore. This is a reminder that all audio associated with this dive will be recorded with the archived video as well as being streamed to shore via the satellite and the internet. Keep that in mind as we proceed with the dive today, and enjoy the dive. Thank you.
Okay, good morning, deep sea explorers. I'm really happy to welcome you today to the first dive of the Seascape Alaska expedition. We're very happy to be at sea today. You know, there's been a bit of a delay, but today we have a bit of a mid-water dive. So I am Rian Waller. I am a deep sea biologist from the University of Gothenburg, and right next to me,
I'm Jennifer Ashoff, and I'm a professor of geology from the University of Alaska in Anchorage. Good morning, everyone.
yeah, good morning. And so before we start here, we're going to have a land acknowledgement before and at the end of each dive, as we're working in Alaskan waters. So as we proceed to explore the deep waters off the Aleutian Islands, we respectfully acknowledge that we are working within and around the traditional Alaskan native homelands of the Sukpiaq, Alutiiq, and Inungun. We acknowledge that the native homelands and honor the indigenous knowledge and heritage of these coastal communities and their relationship with the ocean. We will work with care not to disturb the Alaska Native uses of the ocean environment, and we look forward to building upon this indigenous knowledge to add to our collective understanding of our oceans. So with that, we will begin Dive 1, and first of all, I'm going to turn it over to our mappers to show a little bit of where we are today.
Good morning, everybody. My name is Shannon Hoy, and I am the expedition coordinator for this expedition, but also sitting in as a mapper right now. So if you can join me on Stream 3, we are going to go over the map, give you some context of where we are, where we're diving today, and then we'll go back to talk about some of the science objectives for this water column dive. So if you can see on Stream 3, we've got a map of our operating area. So we left Kodiak yesterday, which is very exciting. We were all extremely excited to be in the water today, and we were able to steam about 21 hours along this arc. Oh gosh, now I'm getting into geomorphology here, but south of the Aleutian Islands. We're right here on Dive 1, and we hope to proceed along the Aleutian Islands as we progress throughout these next 10 dives, weather permitting. So I'll zoom in a little bit now on the bathymetry that we uncovered last night and what we've targeted to be able to dive in the water column over today. While I zoom, if you guys want to talk a little bit about what we're focusing on, I think that would be great.
Sure. So today for water column dives, we're going to be doing different transects within the water column. So just to reiterate, this is a water column dive, which means all of our diving today will be within the water. So we will not be looking at the bottom. We will not be looking at the seafloor today. So to do these water column dives, we have a series of standard transects that we do. Our standard transects are 900, 700, 500 and 300 meters. And within those transects, we hold the ROV steady and we move through the water column at a very steady pace for about 30 minutes or so today. Now today we're lucky enough to be able to do an additional transect to this as well. We're actually going to be working in an area called the benthic boundary layer. And so this is the layer of water that sits directly above the sea bottom, above the surface. So we'll be flying the ROV at about 50 meters off the bottom. So we won't be able to see the bottom, but we're still going to be focusing on the animals that live within the water column. And why this is of interest is because there are very, very different animals that live really close to the surface, close to the seafloor, than the animals that live up in those water column transects. So Shannon here is showing the map of the area. So this was a new map that was generated overnight by the amazing mapping team we have here on board Okeanos. And so what we're looking for today, because we're in the water column and we want to get that benthic boundary layer sample, is we're looking for a flat area where we're not worried about the ROV bumping into anything. We're not worried about meeting seafloor features. And so the mappers overnight identified this little basin up here that Shannon is showing on the screen, on screen 3. And this is where we'll be flying the ROV today.
And for those of you joining us who are interested in the geology, today's dive is right near the continental shelf margin, which is a very important feature in this region. You can see that broad, shallow region just to the north of that mapping area where Shannon is showing her mouse cursor going along. That is the edge of the continental shelf, and you'll see there's a steep break that's just below that. That's our continental shelf break going down into the basin, and eventually we would run into the trench. And so today we're not focusing on geologic features necessarily. We're doing what's called a water column dive, and for those of you that aren't familiar with this kind of dive, the water column, as we use here, is essentially a concept in oceanography that describes the temperature, the salinity, and the distribution of organisms in that column, and as a function of depth. And so these types of dives are really important for gaining information about how the salinity might change, how the temperature might change, and how the distribution of organisms might change. And we also, in some cases, have the ability to collect other information about the water column, like dissolved ions that could tell us about nearby seeps. So we can actually measure, in some cases, dissolved oxygen, dissolved iron, magnesium, manganese, methane, hydrogen, and so this can be an indicator of nearby seeps. So there are a number of different reasons why we study water columns, and as Rhiannon alluded to, there's a lot of different organisms, and they're distributed differently in the water column. And one thing I learned from Rhiannon on this cruise so far is just how the distribution of the organisms changes sometimes depending on the time of day. When you have light coming in at different times, some organisms will migrate to the top of the water column and then later go back down. And so we don't really know that distribution. So from a geologist perspective with very little biology background, that's what I've learned so far. And so I hope you will all enjoy exploring the edge of the continental shelf today, looking at the water column.
Thanks, Jan. Yeah, there's many reasons for this dive today that are really exciting. And on the phone line, I think we have two participants are on the phone. Do you want to quickly introduce yourself, and then we'll go into a mode where the pilots are going to set up for the day.
Mike Vecchione here. I'm a squid guy, actually. I work for National Marine Fisheries Service, which is part of NOAA, and I work at the Smithsonian Institution's National Museum of Natural History, where I'm the curator of cephalopods. And actually, if you don't mind, I'd like to add a couple of things about the reason for doing water column work, if we got time to do that.
Yeah, I think we can take a couple of minutes here. As we get closer to the bottom, the pilots will need time to set up for the transect.
Okay, all of that was really good introduction to the water column, but one thing to keep in mind is that the water column, the open water above the bottom but below the surface, is the largest living space on the planet. Depending on some assumptions you make about calculating volume and so on, it's over 1 billion cubic kilometers of space, and it's somewhere between, well, above 90% of the living space on the planet is the deep sea pelagic area, the water column that we're exploring today. And it's also the least explored part of our planet. So most of the planet has never really been explored at all. And we're trying to alleviate that a little bit by getting as much water column observations in as possible. So I'll get off my soapbox now and let somebody else introduce themselves.
Thanks, Mike. That's helpful. Elena, are you there?
I'll go now. Yes, thank you. My name is Elena Jorgensen, excuse me, and I work for the Alaska Fisheries Science Center with NOAA, and I've been sailing around these parts that we're surveying today for about 25 years, and I'm really excited to see all the animals alive and in real time. So I'm also a squid and octopus expert, but my expertise started with the larval stages, which are going to be found in these midwater toads. So I'm really excited to be here. Thank you.
That's great. Thanks so much, Elena. Okay, I think with that, unless mapping has any more to add, we will go into our dive. So we're going to be a little silent here while the pilots set up 50 meters from the bottom. And then once they are ready to go, we will begin this dive.
All right, stand by one. Let me collect all this info.
Science, this is video.
Hey, Roland, go ahead.
I was thinking about changing up the multi-viewer to be what I would believe would be most useful to mid-water scientists. If that's okay, I'll give them multiple cameras plus the either the vehicle or high pack. I'm not sure which is most beneficial to them. Your mic's not open if you're talking to me.
Whenever you guys are ready. We're ready? Okay. We will officially start our first midwater transect at a depth of 2158 in 5, 4, 3, 2, 1.
Thank you, pilot.
Hey, Okeanos, Mike Vecchione here.
Hi, Mike. How are you doing?
I'm doing okay. Let me turn off my my speaker on my computer. Try to. We've got Keating gnats in the air. Well, we're zooming on a Keating gnat right now. That's an arrow worm for the audience. It's a phylum of worm-like animals that are almost entirely pelagic, which means they spend all their time in the water column. There's a few benthic species, bottom-living species, but almost all of them are up here in the water column. And they're very strange worms, very voracious predators.
Mike, could you say the name of the worm again for me, please?
It's a Keating
A Keating gnat or arrow worm.
Yeah. I think somebody else should
It's
be
like
doing
we're
it.
zooming on a shrimp-like animal. That might be a lophogastrid, or it's hard to tell right now. And I'm not really a crustacean person, but once we get somebody to look at it, we'll be able to get a much better ID on it. I think that's actually a shrimp rather than a lophogastrid. Dougal Lindsay, who's on the chat, says it's a decapod shrimp. He also tentatively ID'd that arrow worm as Euchronia hamata. So we're seeing some cool stuff down here close to the bottom. This is a ctenophore, a comb jelly. There's several kinds of comb jellies, but one interesting thing that's come up about these recently is that increasing scientific evidence indicates that these are what we call the sister group to all the other multicellular animals. So this is a very primitive type of animal that split off of the animal evolutionary line very early. You can see it swimming by beating rows of cilia. That's why they call it a comb jelly. Those cilia rows are called comb rows.
Thanks, Mike. Yeah, the other thing that I really like about ctenophores is we think of jellyfish as being fairly passive, but we know these ctenophores actually travel. They're actually predators. They're quite voracious predators. They go and chase animals and to eat them, whereas jellyfish just kind of catch whatever they come along against. I always think that's kind of neat.
Dougal has mentioned in the chat line that the black pigment patches on this species are different from the North Atlantic morphotype. So it's interesting. Dougal is a jelly expert on the gelatinous groups, including ctenophores and jellyfishes and such. There's another heating nap arrowworm going across the screen.
Oh, it looks like we have a chimera here.
And the first zoom on a fish. This is a rat-tail fish.
Yeah.
These are typically bottom-living fishes, but this is up off the bottom. And sometimes you see them way up off the bottom, but they typically cruise along just above the bottom.
Yeah, it's neat. It's a good reminder that this transect is... Yeah, it's a good reminder this transect is just 50 meters off the bottom, so this is quite a deep midwater transect in what we call the benthic boundary layer. So this is a layer of water that sits just above the benthos, above the bottom.
Right, and it's enriched with both the pelagic things in the water and also the benthic things that can swim up above the bottom, like this rat-tail fish does.
Now, Mike, looking at this image here, the video that we're getting, it seems like there's a lot of stuff in the water from what I'm kind of used to seeing at these depths. I mean, we're over 2,000 meters, 2,160 meters right now. You know, is this normal? Would you think this is normal for this area?
I've never worked in this area before, so I don't know if it's normal. There is a lot of marine snow in this layer here anyway. Marine snow is really important because it's part of the process of transferring stuff from the layers of water that are higher up and sinking carbon and other stuff that's attached to the mucus particles and transporting it all down to the bottom, which is important for moving stuff out of the water column, but also important for feeding the animals that are living in the bottom. And that wiggly thing that we just saw was another arrowworm. There seem to be a lot of them here.
Yeah, I was just going to say the same. There seem to be a lot of those arrowworms. I see a few swimming by occasionally as well. So, yeah, the marine snow is super important.
Yeah,
you know,
it's easy to get confused between those and fishes.
Very true. Yeah, in the way they move, for sure. Yeah, I always think of marine snow as being incredibly important. As a benthic biologist, I'm someone who works with animals that live on the bottom, but in particular what we call filter feeders that are taking particles out of the water to feed. And we always think of this marine snow as incredibly important for feeding those animals that live on the benthos. And so it's really quite amazing to see. You know, I often do work in these kind of depths, and I don't think I've seen quite such a thick marine snow layer before at these kind of depths.
I've seen this kind of marine snow activity in other places, but I don't know how typical it is for here. When we were diving on the Mid-Atlantic Ridge, we got very dense concentrations of marine snow.
Yeah, so is this a Haliceros jellyfish here?
I'm really excited about the video.
Botronyme.
This is a Medusa jellyfish called Botronyme, I think. Excellent zoom. Really nice video. It's one of his targets, Botronyme brucei. It may be one of the collection targets on your list.
Yes, the pilot's going to try and set up and see if we can collect this one.
Cool.
Could you confirm which cylinder that's in?
I'm sorry?
that went into jar number 1, and just for shoreside to realize stream 4 will have 4 cameras, including the sample if we get it.
I don't think I have stream 4 available here.
I'm sorry, stream 3 with 4 cameras.
Oh, okay. Okay, we're seeing more of these rat-tail fishes and some more of the red shrimps, I think. And if you want to log that jelly, Dougal has put the genus and species in the chat.
Yeah, I've got that. Thanks so much, Mike and Dougal, and thank you so much for the pilots for being so fast there. We appreciate it. We're still catching up on data here.
Might point out to the people listening on the internet that I'm located in Maryland. Elena is located in Washington State. Dougal is located in Japan, and we're all collaborating closely together, all participating in this dive off of Alaska. It's really amazing that we can do that. Looks like another Keatignath there.
All right, I think we've just about caught up with our paper keeping back here. So if you're watching online there, the pilots up front just collected a jellyfish, a fairly rare one, and might be characteristic of this area. And that was identified by some of our scientists on shore who are participating in this call. So on board here, we just have 2 scientists. We have myself, Rian Waller from the University of Gothenburg in Sweden. And next to me, I have Jen Ashoff from the University of Alaska in Anchorage, who's a geologist. And we really rely on the scientists on shore to help us identify animals along the way. We have about 300 or so scientists who are interested in this expedition. And each day a different subset come online and chat to us on the chat to help us identify the animals and look at areas of interest. So this is a very wide-ranging exploration, and we have scientists from all over the world, as Mike was saying, participating in this research cruise right now. So we are just finishing our deepest transect. So just as a reminder, this is dive number 1. This is the Seascape Alaska 3 expedition. We're on dive number 1, which is a midwater dive. We're currently about 60 or so miles from land, and we're diving in about 2,160 meters of water right now. We'll be doing different midwater transects during this dive today. So this is the deepest transect that we'll do, that we're coming towards the end of. And then we'll go up to 900 meters, then 700 meters, then 500 meters, and then 300 meters. And the objectives of this dive are to look for midwater animals, so taking video at transect lengths of about 30 minutes or so. And then also trying to collect a few representative species or rare species that the shoreside scientists identify for us. And the scientists, of course, on the phone calls as well.
We've got a request, if we have time left, to zoom on one of the Keatignaths again. The bioluminescent organs of the Keatignaths are visible, and also they appear to have full gonads, so they're getting ready to spawn. So both of those are interesting points. Dougal also was wondering whether both the Keatignaths and the ctenophores might be eating what's called diapausing copepods. Copepods are very abundant crustaceans. There's one of the ctenophores again, getting close to it.
Yeah, this one's a little interesting. It's got the red.
These copepods are... the red is where the gut is of the ctenophore. Dougal is saying that's Volenopsis infundibulum. And I was talking about the copepods. They go through life history stages where they basically stop growing for a while, and they hang out in deep waters until conditions are right for them to start growing and shedding their exoskeleton again. And when they do stop growing, that's called diapause, and it's a life history strategy that helps them get through the bad times and make it to the good times, basically. So we're zooming on a ketognath now, as was requested. The gonads, on this ketognath, the tail part is up and the head part is down. They don't have any eyes, but they have really nasty jaws down at that head part. And the gonads are down towards the tail part where it's swollen out a little bit there. This one actually doesn't look like it's very full yet, but some of the others did. Google's wondering if this is infected by something. All the little tiny dots all over its body.
Oh, this is like a little copepod. Oh, no, a ctenophore, another ctenophore. There we go.
Yeah, that's a different kind of ctenophore. That's a sidipid ctenophore, which has two really long tentacles. It stretches out sort of like long-line fishing gear because it's got dropper pieces that come off of the tentacles. One difference between the ctenophores and the true jellyfish, the jellyfish have stinging cells on their tentacles, whereas ctenophores have sticky cells on their tentacles. So when they catch something that they want to eat, the animal, usually it's a copepod, will stick to the tentacle rather than be stung by it. Google's asking for a zoom on this if you can get close to the body part.
Yeah, I think they're trying to get in there.
The little dropper tentacles are called tentilla, and they're coiled up on this one, so it could extend them much longer when it's actually feeding.
Yeah, so those tentacles extended only when feeding or in different currents? Like, how are those controlled with the ctenophore?
They basically just reel them in when they're trying to swim fast, and then they extend them out when they're fishing, basically.
Oh, really interesting.
They put out a sort of, it's not a net actually. I think it's a lot more like a long line where you've got a main line and then dropper lines off of it.
Whoop, off it goes.
Yeah, Dougal's put a species name on that now in the chat. I'll read it if anybody cares, but it's Okeanos dunlapi.
Yeah, that they have striated muscle in the tentacles. Thank you, Dougal. That's really helpful.
Oh, I see. I missed a note from Elena Jorgensen where she put a tentative ID on the fish. She thinks it's Coryphaenoides cinereus, which is a species of rat tail.
It's either another one or the same one of these ctenophores.
Yeah, I think it's another one of the same.
Euplacomus. Fishing for lunch, Dougal Lindsay says. It's has its tentacles spread out beneath it, ready to catch some food. Oh, let's realize we're there.
You see it reel in its tentacles when it gets ready to swim.
Yeah.
It's got one of the two tentacles reeled in really tightly, and the other one's still extended a little bit.
Off it goes. Yep.
And this, in the background, is a different type of ctenophore. It's called a lobate ctenophore. And classically, when you study biology, you learn that ctenophores swim by using these rows of cilia to beat the cilia. They set up currents that move the body along. But lobate ctenophores are actually capable of frog kicking with the lobes that come off of their bodies.
It's on.
It's a Bolenopsis. This one right now is swimming with with the comb rows, but it's capable of using those lobes that are dragging behind right now to actually swim sort of like a frog kick. Dougal pointed out that ctenophore had parasites on it. And that's an important thing about this pelagic world, that a lot of things live on jellies, and some of them are parasites, some are just hitching a ride, and some are actually eating the jellies as they live on them. It's another ctenophore, comb jelly. The animals that live in the water column many of them are gelatinous, and many of them are in these groups that the comb jellies and the jellyfishes and other gelatinous groups like salps and so on. And because of how big the pelagic environment is, the open water environment of the deep sea, the numbers of these things are really huge, even though you don't see a lot of them all packed in together. When you multiply it out by the volume of water that they live in, it's huge numbers of them. And when I teach deep sea biology, I tell my students that if an alien civilization came and took a random sample of life on Earth, what they would get is a gelatinous animal from the deep pelagic. Dougal's pointing out that some of these ctenophores have the black pigment on them, and some don't, which is it makes it a little more difficult to figure out what it is we're looking at.
Do we know what that black pigment is for, Mike? Or Dougal?
No, I think Dougal was thinking it might be parasite.
Oh, interesting.
His text was no idea.
No idea. Yeah. Like so many things in science, we really don't understand a lot of these animals living in the deep ocean.
Well, this is what exploration is really about. We are exploring this poorly understood ecosystem, and as we're exploring, we're finding out new things.
Yeah, those black spots seem so regular. It'd be hard to believe they were parasites.
Dietary.
Yeah.
Yeah, they do look very, very regular on that animal. Many of these gelatinous things that we've been seeing are individual animals, but probably later in the dive we'll see some others that are colonies, because it's very common to see colonial gelatinous animals as well up in the water column.
Yeah, and it's that gelatinous nature that makes them able to survive in the deep ocean very often. If you have animals with bones, the deep pressure at these depths could crush those bones and crush those air spaces, and they just become very, very heavy and hard to move. Whereas you're gelatinous, you have what's called a water. You're adapted to living in water. Most of your body is water, and so you're not affected by the pressure quite so much. We have an interesting red jelly.
Right, and in addition to that, a lot of them will replace some of the ions in the fluids in their bodies with ions that have the similar charge but are lighter, so it actually provides buoyancy without it being a gas that would be compressed. I'm seeing a really small sidipid ctenophore go off the screen, and there's another ctenophore in the background there, it looks like.
It's like quite a bit larger one, or is it a mucus net?
Yeah, Dougal just pointed out if you have bones that are exposed to the water, when you go down deeper, the calcium. That's a house for.
Is it larvation?
Larvacean.
Yeah. These larvation houses are really interesting because they're primarily mucus. The larvation itself is a really tiny animal that lives right in the center. I don't know if we can zoom in there and take a look at it.
Yeah, I don't see the larvacean in that one.
I think he's right in the middle there.
Often, if it's there, you can see. Well, that might be. I don't see the tail to it. So usually, you'll see the tail beating inside, setting up a current. But
oh!
Dougal says, "Yeah, that's him."
Oh, abandon the house.
Oh.
Off it goes.
Now you can see the tail flapping. Yeah.
Yeah. So these larvaceans can build these mucus
houses. They're more closely related to us than they are to the jellyfishes and such.
Yep.
Very cool.
Oh, he's abandoned.
And as it abandons ship, that's a mucus structure that it builds to feed. So when it's in it beats its tail to set up a current to push water through the mucus and filter out particles in the water. And they eat very tiny particles that they filter out of the water. But another important thing is when they abandon ship like that, they leave behind this mucus with all the stuff attached to it. And that is a major contributor to the marine snow. But also a lot of times it will sort of crumple up together and will sink faster than the tiny particles will. So it transports carbon out of the water column and down to the bottom where the carbon can be eaten by benthic animals. But also it's taking carbon out of the carbon cycle that's affecting the climate on the planet.
Yeah, that was really neat to see that.
That those when the stop houses are abandoned, they can sink up to 1,000 meters per day.
Yeah, I know there's certain areas of the globe where those mucus houses, those mucus nets, have been shown to be incredibly important for the benthic and food environments, so bringing all that really dense food down to the seafloor, feeding the animals that live down there. And they can build those mucus nets several times a day, from what I remember. Is that right, Mike? They can build them within a few hours?
They can rebuild them pretty quickly. I don't know how often they do it. Dougal says that the houses provide 50% of the benthic need for food. What we're seeing here is keating gnats, these arrow worms.
Yeah, lots of keating gnats today.
There are a lot of keating gnats here. They also feed on copepods.
Oh, I think here is another one of these mucus houses, maybe.
Could be in the background there, yeah. Usually there's an inner house that's easy to see and then an outer mucus structure that's very difficult to see. And both function together to trap it. Yeah, you can see the tail of that larvacean beating inside that mucus house.
Oh, that's a beautiful shot. Look at that.
So what it's doing is pushing water through the filter so that it can trap marine snow and other particles. They feed on very small particles. And
you can just see all the particles in that mucus net right now.
Mucus, they can eat it. Yeah, and once the mucus filter gets clogged up with marine snow and stuff like that, they will abandon it, just like we saw, and build another one that's not clogged up.
Yeah,
But you can see how complex that structure is.
Dougal Lindsay on the Science Chat here is saying they actually have another one of these houses already. They keep one ready to inflate under their chins, so as soon as they abandon their house, they can build another one almost instantaneously. That's a beautiful shot. Look at that. Yeah, you got two large eyes and a
Very
nose.
nice zoom.
Yeah.
All right, everyone, this is the official end of our first transect this morning.
Thank you so much, pilot. Okay, so that's the end of our deeper transect. So now we're going to be moving up to 900 meters water depth to take another transect up there.
Okay, we're moving up into what's called the mesopelagic, or mid-level depths, the twilight zone. Some people refer to it. So we'll be doing the rest of the work up in the twilight zone, which is a really interesting area. And thank you for getting a deep transect in.
Yeah, that was really interesting. You know, I'll be intrigued to see what different organisms we might see in that slightly shallower depths.
I'm going to put my phone on mute until we start seeing things I can talk about.
So on the quad split, I put the normal dive screen up so you can see our depths and their location on the high pack.
So if you're just joining us today, this is the first dive of the Seascape Alaska 3 expedition. We are doing an Aleutians water column dive today. We've just completed our deepest transect, which is in what we call the benthic boundary layer at 2,160 meters. And we're, at the moment, just moving up through the water column. We're going to be taking another transect at about 900 meters. So this is dive number 1 of the Seascape Alaska 3 expedition.
Hi everyone, this is Jennifer Ashoff. I'm the science lead in geology. If you're just joining us, we are on a dive that is about 60 miles near Tugiak Island, and we're ascending to a water depth of about 1,960 meters. And we are beginning a dive where we're completing a series of water column transects. So as we move up through this water column, you'll see a wide range of ctenophores and various other jellyfish. I'm a geologist. I'm doing the best I can here. But the goal is to take samples of the water as we move up through the water column. So if you're just joining and you're seeing a lot of particles moving by your screen, that's what you're seeing is marine snow and a wide range of jellies. And so once we reach 900 meters or so, we'll complete another transect and take another sample. And at that point, we'll take some time and discuss some of the organisms that we encounter at that water depth.
Yeah, that's a really good point, Jen. I don't think we've talked about yet that we're taking these water samples. So at each of our water depths, we're taking what's called a Niskin bottle. And this is a bottle that's on the back of D2. And they have lids that are open right now. And then when we want to take a sample, those lids close and take a sample of the water. And what we're using that water for is a process called eDNA, or environmental DNA. Environmental DNA is a fairly new technique where all of the DNA in a water sample is taken out and extracted, and then they're able to tell from that what organisms are either in this water mass or close to this water mass or have just traveled through this water mass. So this fantastic new technique is giving us really good insights into what we call biodiversity, so the types of organisms that live in areas. And the theory is that we might be able to use this eDNA in the future to be able to go out to sea, just take a quick water sample and be able to say, okay, these kinds of fish live here or these kinds of corals live here, without having to deploy more expensive assets to be able to do it. It will give us a much better idea of where animals are in the ocean. So that's eDNA, environmental DNA, and we take those samples at each of our transects.
Rhiann, since we have a little bit of time, I was wondering if you could explain some of the creatures that we've observed today and some of the differences. For example, what's the difference between a ctenophore and a jellyfish?
Oh, now you've got me. Mike might be the better person to tell. But yeah, so a jellyfish is in the phylum Cnidaria, and ctenophores are in a different phyla. And they're gelatinous animals. They have a lot of similarities. They look fairly similar. But the big difference that I know between the jellyfish and the ctenophores is these jellyfish are these passive swimmers. So they have this swimming bell, and they have a little bit of movement. They can orient themselves a little bit in the water column, but they're mainly moved by currents. Whereas ctenophores have these comb rows that Mike was telling us about earlier. They're called teens. They're the bits that kind of look silvery, iridescent when they swim. But they're actually mobile, so they can move through the water column. They have much more control over where they are. They can move, and they're actually predators, so they will go and chase prey, whereas a jellyfish kind of relies on whatever comes past its tentacles to be able to snatch them. Some other differences between the two is the Cnidarians have stinging cells, little harpoons that come out of their tentacles, and ctenophores have something similar, but instead of being harpoons, they're actually sticky, so they stick around prey instead and kind of capture that prey. So they have a lot of similarities, but a lot of differences too. Both can be bioluminescent in some ways. Most can be transparent, or they can be different colors. We can see some red jellies and some red ctenophores. We can see clear ones of both. So there's some similarities and some differences.
Okay, that's really interesting. And how do the ctenophores sense their prey?
Yeah, they have special little sense organs inside the ctenophores. I don't actually know too much about it. I don't know if Dougal's going to give you a bit more information here online. He is the main jelly person. But yeah, from what I know, they have little sense organs, and so are able to sense prey. Probably not very far from them. So again, kind of waiting for what's around. But yeah. So Dougal Lindsay, who's on the science chat, is saying smell, so they can smell their prey. So, really interesting.
Thanks, Dougal. That's really cool.
Yeah, it's a good time to remind people that, it's myself, Rian Waller, and Jen next to me, Jen Ashoff. But there are many, many more scientists on shore who help us out during these dives. We have a science chat here where scientists from all over the globe can talk to us while we're online, help us identify animals that we're seeing, identify processes, identify rocks and geological features. And so we're far from doing this alone. We have this great team of scientists who are on shore as well helping us out. As we're moving up through the water column, getting to our next transect, which will be at 900 meters. We're seeing lots of jellies in the water, lots of tentacles, more of those arrow worms. So we're seeing a lot of things going by at fairly high speed. There's another one of those larvation houses. So even though we're moving between the transects here, even these video from between transects is still incredibly valuable for understanding the biodiversity in this area. So what kinds of animals are living in this area? A little fish, myctophid. There certainly seems to be a lot of life in this area. And just to remind folks, we're off of the Aleutian chain right now. We're not too far from the Aleutian Trench, which is a subduction zone, an area that's very deep in the ocean. We are currently about 60.5 miles or so from shore. We are in 1,600 or so meters of water, and we're aiming for the 900 meter range. And we're in the middle of dive 1, which is a mid-water dive that we're having. So we're looking at animals that live in the water column. So we've just completed a water column transect in what we call the benthic boundary layer, so really close to where the seafloor is, and about 50 meters off the bottom. And now we're coming up to do a transect at 900 meters. And once we've completed that one, we'll also do one at 700, one at 500, and then one at 300 meters. And the reason that we do so many different transects at these different depths is that different organisms like to live in different parts of the water. I think traditionally people think of the water column as kind of this big mixed mass, that it's all the same, that it's not very different. There's lots of different currents in the ocean, some currents that run deep, some currents that run kind of in the midwater, and some currents that run in the shallow water as well. And they have very different properties. They can be different temperatures, different salinities, different oxygen levels. And so different kinds of animals like to live in these different areas. And then today we were fortunate enough to do a dive down at the benthic boundary layer, 50 meters off of the bottom. This is an area that's very, very understudied. This area can have animals that like to feed on the seafloor, that kind of come up off the bottom a little bit and then go down to feed. So it can have a very different fauna, a very different composition of animals than what we'd see up in the water column. So we're hoping to see some different things in this 900 meter transect that we're coming up on in the next kind of 15 minutes or so. We should get there. It's like a bloom of ctenophores here. Look at all the ctenophores in this video. More and more. I wonder if these are the same species that we've taken video of earlier, some close-ups. They certainly look very similar. They're those low-bait ctenophores, so not the ones with the big trailing tentacles, but the ones that have almost wings that can come out and let them frog kick to swim, as Mike Vecchione was telling us earlier. In shallow water, these ctenophores can sometimes bloom in such high numbers when the conditions are correct. They can clog ships, ballast water, and clog up shipping lanes. Dougal Lindsay has described this as a Bolenopsis party, and I quite agree with that description. I think it is a little bit of a party going on in this layer. And we're sitting right now in about 1,400 meters of water or so. To add to the ctenophore party, Dougal Lindsay is saying the way they iridesce, so those teens, those comb rows on the side have this kind of iridescent glow to them, and they look a little bit like a disco ball, a little underwater disco. Though interestingly, we seem to almost be out of it now. Not quite so many ctenophores going by. Oh, a little siphonophore. Yeah, still lots of life in this layer. Another. Oh, and we're back into ctenophores. And the siphonophore just went by. Party number 2. Oh, beautiful. If you are just joining us today, I'd like to say welcome to the first dive of the Seascape Alaska 3 expedition. We are working off of the Aleutian island chain, and today we're on a water column dive. We're currently about 60.5 miles off of land, and we're working, our deepest depth today has been 2,260 meters. And we're currently on our way up to do a 900-meter transect. So for these water column dives, we do transects in different depths of water. We started out our deepest at the benthic boundary layer, and we're moving up to 900 meters. Once we've completed our 900-meter transect, we'll move up to 700, and then 500, and then 300 meters water depth. And what we do in these transects is we stop the D2, the ROV, let it settle out, and then they drive forward at a slow pace for about 30 minutes or so. And during that time, we take video images, and then there may be some collections as well during that time. And these videos are incredibly valuable for scientists to be able to look at the kinds of animals that are living in these different layers. The water column is far from a mixed, homogeneous place. There are lots of different climates, lots of different habitats within the water column. The animals that live down towards the benthic boundary layer are very different from the animals that live up in the water column. So we're hoping to see some different things in each of these transects today. So we probably have another 10 or so minutes here before we get up to our 900-meter transect. Then the pilots will settle out the vehicle, get everything nice and steady, and then we'll begin our transect at 900 meters. You got that right, pilot. Solonimissus. It's a pretty dinner plate jelly right there. Not quite in the transect, but still, it's a beautiful specimen. They're called Solmissus is the genus name.
That for Dougal?
I think we're just setting up Mike, so I don't think collection's on the cards right yet because we just got to this transect. So that's beautiful. One of the features of this jelly is the tentacles are often facing upwards rather than downwards.
They always look like a flying saucer to me.
Well, he wants to grab this. I don't know if you have enough time to do everything. Yep. This is the official start of the second midwater transect for the day at 900 meters.
Dougal has asked for a zoom before collection. Zoom on the stomach pouches, which I think are around the edge of the bell on this thing.
Ready on science.
Oh, George Matsumoto just joined us, too, on the chat. Another expert on jellies. Nice zoom.
This is beautiful video of this. These are called dinner plate jellies of the genus Solmissus. This type of deep sea jelly, one of the features is its tentacles actually face, a lot of the tentacles face upwards.
I think Dougal's satisfied with the zoom. If we could go ahead and slurp it.
Okay, attempting to sample.
Uh-oh. That looks like a maybe not.
Yeah, it looks like it's going to be too big for the suction. No. I don't think so, no. No, the stick with suction. Yep.
If we get any pieces in the jar,
Thank
we
you.
can get DNA from them.
Yeah, if we can just get a tentacle or two, which you probably have, so we should rotate the jars. Yep. Oh, yeah. There it went. Okay, collected. Definitely have pieces.
DNA sample to me.
Yeah. Can we confirm that went to canister 2? Thank you, pilot.
And there's another one. I don't think we need to spend much time on this one. The sharp, though, might be interesting. It's like a mysid. Ah, there it goes. Yeah, Dougal says that's the same species. We can continue on the transect.
We're getting some still images that you guys can't see, or a different camera we're processing.
This is the pilot speaking on OKEX right now. It may look a little odd in our main screen. We're working with a new camera that's recently been installed on the vehicle, getting some high-resolution, high-definition stills of this jelly. And we will be moving on just momentarily.
Beauty shots.
Yeah, if there's any way to paint the lasers on that, that would be helpful. I don't know if it's too high up. It might be.
All right, well, let's come.
Thank you, pilots.
That shrimp looked like a mysid. It's called a possum shrimp. They have a brood pouch underneath their carapace, where they refer to them as possum shrimps.
A little myctophid fish there, maybe?
Yeah, let's get a zoom on the fish.
Oop.
Looks like a myctophid to me.
There it goes. So if you just joined us, this is just a reminder that we're on dive 1 of the Seascape Alaska 3 expedition, and we are currently off of the Aleutian Islands diving a midwater dive, so looking at animals that live in the water column. We've just completed our deepest dive, and we're up now at 900 meters, and we just started a transect looking at different organisms.
Yeah, and for the people who are listening at home, 900 meters is the deepest part of what's called the mesopelagic, or the twilight zone. And it's called that because, at night the whole ocean is dark, but during the day sunlight can penetrate into the water, and it can penetrate globally. The sunlight can penetrate down to about 200 meters with enough power to drive photosynthesis, the chemical chain that plants use to turn sunlight and carbon dioxide in water into food. But below 200 meters, there's not enough light to do that. So that's part of the reason it's called a twilight zone. But sunlight can still penetrate deeper than 200 meters. And again, globally, down to about 1,000 meters is where the light finally gives up. And, you don't get any more sunlight below about 1,000 meters. So this twilight zone is from about 200 meters to about 1,000 meters. And a lot of interesting things happen here. This is where the biggest migration on Earth occurs. That's where every day lots and lots of animals swim up towards the surface and spend the night up there feeding, and then when the sun comes up, they swim back down into the deeper mesopelagic. So it's a dynamic area. It's not as big as the bathypelagic, the layer below it, but there's a lot of stuff going on, and there's more abundant animals in the mesopelagic in general than the bathypelagic. But you'll notice that here we're not seeing as much stuff as we did in the first transect, but that was a transect that was in the benthopelagic. That's the layer that's right up against the bottom, and that's, like we were saying before, that's an enriched layer. So we're just starting to work in the mesopelagic here, the midwater or twilight zone. You might notice also there doesn't seem to be as much of this marine snow here as there was in that first transect area. The marine snow is just sinking right through here, going down to the bottom. What we're looking at here is a true jellyfish, as opposed to those home jellies that we were looking at before. This is one of the ones that has stinging cells. That's a very nice zoom on a very small animal. Perfect. Dougal says this is Pentacogon Haeckelii, named after Ernst Haeckel, a very famous jellyfish biologist and artist. George Matsumoto is pointing out that this has got really nice gonads. Are we allowed to say that on the internet?
We are asking the community in the chat, just to double check.
This is Watch Lead, and no one in the chat has indicated that this is a sample target. Thank you.
It's a very tiny red enophore. Red is a very common color at these depths because what light is down here is blue light. Red doesn't penetrate through water very well. Oh, Dougal is saying that might be an undescribed species of ctenophore. The reason that red is a common color is because the blue light that is all that's common down here does not reflect off of red pigment. This is a doliolid. It's related to you and me, actually. It's south. Well, it's related to the south anyway. It's called a nurse. It's budding off a whole bunch of babies. So these are all clones of the big one that's off to the right. So it looks like a tail there, but it's a chain of buds that are clones of the original animal. These things only live to reproduce, like Dougal is saying in the chat. There's no stomach. They can't feed. There's a very nice image of that doliolid. Something spherical in the background there, too. Possibly one of those Gran Rojo jellyfish. Hyperonia Gran Rojo, the big red. Oh, nice. Nice zoom on the oleola. hard to get a good picture because they're so transparent. Hard to get the lighting on them correctly. Oh, there it goes. They're jet propelled. Google says George named it. I think he's talking about the jellyfish, actually. George Matsumoto, who is on the chat, I think was one of the people that described it and named it. Tiburonia, named after the ROV that Monterey Bay Aquarium Research Institute uses in Gran Rojo because they call it the big red jellyfish. I see a copepod. The copepod's drifting above it. Somebody mentioned that there was a mysid on it. Very nice. If I remember correctly, these things don't have the stinging tentacles like other jellyfish do. They have what are called oral tentacles, but they don't have the tentacles around the edge of the bell. Am I saying that wrong, Google or George? Only oral arms. Very strange jellyfish. No marginal tentacles. You can see this, the bell has been damaged in a few places, and if you look closely on a lot of these gelatinous organisms down here, you'll see amphipods and other animals riding around on them, but they're also eating them at the same time. So they're hitching a ride, but they're chewing on them as well. Google's saying that these have nematocyst warts. I don't even know where those are. I'm not a jellyfish person. The spotted appearance there are nematocyst warts. That's what Google says. I've learned something today. Very nice. Google says the jelly is so massive that the radial canals need to form a network in order to get nutrients to every corner of the jelly, the body. I think we've got enough of this. I know Monterey Bay Aquarium Research Institute has got a lot of detailed observations on this species. Google says that's good for AI, lots of angle. What do we got in the background there? Looks like a siphonophore, a Physonex siphonophore. Bargmania, possibly. There's several different kinds of siphonophores. Oh, in the foreground, that looks like squid ink, actually, that yellowish stuff. I started to say that most of the siphonophores we see down here fall into two groups. One is this kind, which are the Physonex siphonophores, which have a lot of swimming bells. This is a colony, actually. There's a whole bunch of animals that are stuck together. They're called persons by siphonophore people. But the Physonex have a little tiny gas bubble up at the end of them. And I've never really understood what the function of that is. That looks like another squid ink there, a pseudomorph. The other kind has just a few swimming bells and does not have the gas bubble at the end of it. That's called a calicophoran siphonophore. I think there must be a squid around somewhere, but it's not cooperating with our cameras. Google points out that the gas in the float on those Physonex siphonophores is carbon monoxide, which is poisonous to many kinds of animals. There's another one of his dinner plate jellies, Solmissus. Zooming on something red there. Looks like it's a decapod shrimp. Again, the red color is very common in many kinds of animals at this depth. You would think that red would not be a very good way to hide, but if the only light around is blue and it will not reflect off of the red pigment, then people can't see it. Not people, the predators can't see it. Dougal's suggesting a name of Benthiogenema, but he thinks the tail was too thin. Ah, I told you there was a squid around. Can't hide forever. This is where Elena Jorgensen's got to get on the line and tell us what it is. My guess is it's some kind of a gonadid. The clubhook squid. This is where Elena's tentacles are being tied together. Elena?
Yeah, hi, Mike. I'm sorry. Yeah, it looks very much like a gonadid, but I cannot tell you the species. Do we have a guess about how big that is?
Oh, it looks like it's got a photophore on the eyes of it.
Oh, really? Is it the ventral eye? Because that would make it a pyros.
Anteroventral, pyros? Gonadus pyros?
Correct.
Well, I was getting a reflection that looked like a photophore on the ventral part of the eye, so that might be what it is. Very nice zoom on it, though. And I didn't take a screen grab. How stupid of me. I'll have to get one from the ship. Nice. Very nice. You see that photophore right on the ventral, anterior ventral part of the eyeball?
Yes, and that... Sorry, I'm just trying to get a good look at it. Yeah, Pyros, as far as we know, is the only one that has the eye photophore in the family Gonadidae. And this does have the right body proportions to be a Pyros. So, oh yeah, and that's a really good shot, really good angle.
So is this a common squid in this area?
Thank you, pilots and videographer.
This is really interesting. This is like a jelly with something hanging out.
Can I ask you more questions?
That's a jelly that somebody took a bite out of.
Is that part of the jelly there? Or is that something else that's hanging off the bottom?
Well, that's one long tentacle. They have a bunch of shorter tentacles and then one extra long one.
Okay.
That's used for prey capture.
The different color there?
Dougal, before I embarrass myself, why don't you tell... Oh, you did tell us what it is. It's Atola.
An atola? Yeah.
And that Atola. And that long tentacle is characteristic of it. But this one looks like it's old and beat up or something chomped on it.
Something had a snack.
Everyone, the pilot has informed us that they are getting back into position, so it'll be just a moment. And while we get back into position, I'll just remind you that we are on the third expedition of Seascape Alaska on our very first dive, which is a dive to investigate the water column. And we're approximately 60.5 miles from Tugadak Island at a depth of around 900 meters. And we're currently completing a transect at the 900-meter mark to explore some of the organisms at this water depth and take samples of the water.
And that looks like a Paralia that somebody chomped on. Or again, I might be embarrassing myself, but it does look like it's been...
Yeah, it definitely looks like it's got a chunk missing out there.
Paralia rufescens.
Yeah, Paralia
Yeah, Dougal
rubensis.
says it's Paralia rufescens. And like the other one that we just saw, something's been feeding on it.
Okay, everyone, this is the official end of our second transect today.
Okay.
Thank you, pilot. Okay, so we're going to be moving on from this 900-meter transect and moving up to 700 meters. Shouldn't be too long between these two transects.
Okay.
And yeah, that was a beautiful squid. What a great shot.
Yeah, Dougal reminds us that he'd really like to get a sample of one of those, the Bolenopsis, which is the lobate ctenophore. There was just one that floated by as we, just like less than a minute ago.
Yeah, we're getting ready for transect here, so we can't capture that one, but we have it on the list here if we see that in the next transect. Yep.
If we get too high up, we might not be seeing them anymore. we started the transect now?
Not yet, Mike. We're just getting set up here, and then they'll begin the transect. They'll announce it when we begin.
Okay. That's a myctophid. Those are very common and abundant fishes. Well, Dougal says it's a bathy lag. It looked like a myctophid to me. That was a shrimp flipping away. There's one problem with working remotely is there's a lag in both the video and in the telephone connection. And we never can tell exactly how much lag there is and whether the lag on the telephone and the lag on the video are equal or whether it's a different lag between the two. So, and the same's true also for the chat room. So sometimes when we are communicating with each other, we're talking about different things and don't actually know it. This is a jellyfish called Paralia. Always looked like a big head of cabbage to me.
They're about to take. Beautiful jelly there.
Yeah, you can see a difference between that and the big red jelly, the Gran Rojo, because this has got tentacles all around the edge of the bell, which the Gran Rojo does not. And the oral tentacles coming out from the center are much larger on the Gran Rojo. And Dougal was asking, he apparently saw an amphipod parasite on that one. Very common to see small amphipods on the jellies.
Excellent. Thank you, Nev.
The colonial jelly we just passed was a siphonophore called Apollonia. I think that's one of the other group. That the name escapes me right at the moment. It's not that it's Pisonex, it's the other group.
Okay, so we're starting the transect now officially. This is the 700-meter transect that we're taking. And if you've just joined us, we're on the first dive of the Seascape Alaska 3 expedition. We are off the Aleutian island chain. We're about 60.5 miles from shore right now. We started our dive today at 2,260 meters depth, and we're taking different transects through the ocean through the midwater. We're looking at midwater animals today, so the animals that live in the water column, not the animals that live on the bottom. So we've done a transect down at the bottom. We did a transect at 900 meters, and now we're starting a transect at 700 meters depth.
And since the mesopelagic extends from 200 meters down to 1,000 meters, we are pretty close, just a little bit below the middle of the mesopelagic.
Oh, the beautiful siphonophore.
Nurse doliolids, which is reproducing asexually, forming buds all along that tail. Google has identified that as a rapallonemid medusa, and there's another one of those dinner plate medusae, the solmissus.
It seems like there's a lot of these solmissus around here. Is this fairly usual, where you see one, you see a lot, or do you usually just kind of see one?
Actually, they're very common in a lot of places, and one thing that Dougal has been working on is trying to figure out what the species boundaries are, I think. I'm speaking for Dougal out of turn, but it is a very important midwater predator. And we don't know how many species there are yet, but because deep discovery are able to collect specimens, then we can get DNA out of them, and Dougal can use that to help figure it out.
Yeah, it's really interesting.
He really wants one with 24 tentacles or less.
We'll see if we can do some counting in the back row here. Yeah. Quite a job to count those tentacles while we're moving through the water column.
The shrimp right up at the top there. What have we here? Oh, it's another Physonic siphonophore.
Is this the fire belt siphonophore?
Spread out there. This is a colony of well-separated kinds of animals. So there's a lot of specialization. There are swimming animals, and then there are stomach animals, and then there are other specialties as well, reproductive animals. Dougal says that's Maris orthocana. So that's a fairly common species. Little shrimp.
Be another little myctophid going by. It's a little tiny midwater fish. This is a little shrimp.
Yeah, that looks like another one of those mysids, the possum shrimps. And Dougal saw a polychaete somewhere in here.
Oh.
Pelagic polychaetes are really cool when you get a chance to look at them.
Again, it's certainly looking like this layer has a fair amount of that marine snow in it. Maybe not quite as much as the deeper layer where we were in the benthic boundary layer, but there still seems to be quite a lot of particles in the water, Would you agree with that, Mike?
Yeah, I agree with both statements that there's a lot in the water, but it's less here than it was down deeper.
Yeah.
Seems to be.
Could that be due to mixing,
There's a
bringing
kind
up
of
some of the
worm
sediment?
here that I've seen a few of. It's called Piobius, which is a very strange worm.
Oh, look at this little jelly. Beautiful. This looks like a hydrozoan jelly. Would that be what that is?
I'm going to wait till either Dougal or George wait.
Yeah. Pentacogon hecklei. Hydrozoan, yes.
Yeah. Yeah.
Yes, there's two main types of jellies that we see, the scyphozoan, the true jelly, and then hydrozoans, which have the jelly as part of their life cycle. But they're primarily benthic-based, often. Not always, but often. Very often the hydrozoans tend to have more of a bell shape.
Pelagic hydrozoans that have their attached stage, they attach to shells of pelagic snails.
Oh, interesting.
The theropods. The attached part of the life cycle is actually attached to a snail that's swimming all the time.
So they attach to the snail for the entire life cycle?
Well, no, just for what you would call the benthic part of the life cycle, the attached part.
Okay.
And then they turn into a jellyfish for the other part of the life cycle.
Oh, interesting. Oh, what's this one here? It's another jelly with a bite taken out, it looks like.
It does.
Another Poralia, maybe?
Yeah, I think so.
So what feeds on these jellies at these kind of depths? Is it other larger fish? Is it, what tends to munch on the jellyfish?
There are fishes that feed on the jellies. Those bites look too big to be coming from something like an amphipod, but the answer to your question is I don't know. But I'm glad something feeds on them.
Something is definitely feeding on these of different species as well. We've seen quite a few with bites taken out.
Yeah. Oh, Dougal suggests maybe deep diving molas.
Oh, sunfish.
We're actually at a depth that the leatherback turtles could dive to, but I don't know that leatherbacks get up this high, this far north in the Pacific. But sea turtles eat the jellies, and particularly leatherbacks dive down into the mesopelagic to feed on jellies.
Yeah, interesting. I wonder if it's too cool in the waters here for sea turtles, but I don't know that either.
I don't know. But mola do feed on jellies.
Yeah, and the mola, those are the ocean sunfish?
Yeah.
Yeah, Elena Jorgensen is saying in the...
I don't think they've ever collected a mola this far north. So, we've identified a mystery.
We have identified a mystery.
Out of this jellyfish.
Something is having a bit of a feeding frenzy on jellies down here.
Well, the owlfish are jelly feeders as well. I should be able to remember the scientific name because I published on them, but I have to look back here. Bathylagids. Bathylagids are jelly feeders.
Okay.
But I don't know that they would take a chunk like that. I don't know. Interesting. another tiny jelly.
Yeah, a little one.
Yeah, Dougal agrees that the mouth is too small on the Bathylegids to take the kind of bites that we're seeing. That little jelly is called Botryonema.
There's another little siphonophore, maybe? Yeah, it just went off.
Doliolid, maybe. Shrimp. Yeah, there's no Bolenopsis here. Those lobate ctenophores that we were seeing, and we're hoping to get a sample of.
No, they were having their disco party down deeper depths. We're certainly looking out for them, though.
There's a Piobius, I think, going off to the left there. That's a worm that does not have segments in its body. And that looks like mucus to me.
Yeah, certainly a lot of mucus makes up this marine snow. Marine snow is really just particles that are coming down from the upper ocean and raining down into the deeper ocean, and they can be made up of many, many different things. Degraded body parts of animals that have died in the upper column, plankton, phytoplankton, zooplankton that's died up in the upper water column, but also sediments as well that have blown into the ocean or washed in through rivers as well. But this marine snow is incredibly important for the deep ocean. It really feeds the deep sea.
The exoskeletons of the crustaceans, as they grow, they shed their exoskeletons, and those become part of the marine snow.
Yeah,
and also there are a lot of animals that use mucus to feed.
This little red guy here. Is this another ctenophore?
On that red thing.
Yeah, ctenophore. Another cydipid.
Oh, yeah, it's a cydipid ctenophore.
Yeah.
A red cydipid.
Another beautiful
A lot
Coralia.
of things use mucus to feed. Some of the gastropods do, and of course the larvaceans make these big complex mucus houses, and other things use mucus as well to feed. And then the mucus becomes abandoned after it's been used, and it becomes part of the marine snow as well.
Yeah, we saw that amazing larvation house earlier, and with the larvation actually bailing out and escaping its home when we got a little bit too close. That was amazing footage. I've never seen that before.
Shrimp. I wonder if one of these shrimps would cooperate for a close-up.
Yeah. They seem to be swimming out of our range pretty rapidly as soon as we get there.
Yeah, they're pretty picky about things approaching them.
With good reason, I think. D2 is quite a bit larger than these tiny little shrimp. I'd be a bit scared.
Yeah, brightly lit up and making all kinds of noises and vibrations. And
yep.
That would be terrifying.
Is that another little myctophid fish, these little midwater fish?
Let's see.
Yeah. Oh, there he goes.
Yeah, I think it is a myctophid. I haven't seen very many bristlemouths, so you usually see quite a few of those.
Oh, another one of these jellies that have the long trailing tentacles. Is it Atolla? Atolla jellies? Yeah, Atolla. There we are, in Dougal. They have this long trailing tentacle that's for feeding.
That one long tentacle is characteristic of it.
Beautiful. That long tentacle looks so out of place in this bright red jelly, having this bright white tentacle. And these jellies will have stinging cells, so they're in the phylum Cnidaria, which is characterized by having these cnidocysts, which are these stinging cells that have harpoon-looking things inside them. And when they catch prey or find something they want to grab onto, they stick out these harpoons. The harpoons grab on. In some species in shallow water, they even contain toxins and poisons.
And that thing flips around. You see there's a ring of white ball-like things around the close to the edge of the body. Those are photophores, and when it lights up, the photophores actually light up sequentially, so it goes around and around and around and around.
Yeah, I think there's been the
thought to be an alarm.
Yeah, burglar alarm, I think is what I've heard that called before.
Yeah, burglar alarm. Yeah. And Edie Witter made a lure that lights up like that and put it in front of a camera, and that attracted a giant squid.
Oh, wow.
And the thought is that the jellyfish does that when something's trying to eat the jellyfish. And when it lights up like that, it attracts a bigger predator, which will come along and eat whatever's trying to eat it.
Oh, beautiful jelly. Coming right up. The tentacles are just kind of retracted. Yeah. Can feel our presence, I think.
Pentachogon hickeli.
And a young one. Yeah.
The gonads, they're not as developed.
Oh, look at that iridescence as it's going upside down.
Yeah, a lot of people will mistake that for bioluminescence, but you're right, it's iridescence. It's breaking up the light that's coming from the ROV. It's the Sidipid ctenophore that went off to the bottom right. And the siphonophore going off to the left. And another big Paralia. Ian, I'm going to have to sign off for just a minute, for a few minutes. I'll call back in a few minutes.
Thanks a lot, Mike. We appreciate it. Thanks everyone who's tuning in, and just as a reminder, this is Seascape Alaska 3rd expedition, first dive, and we are diving in an area that's about 60 miles off of shore of Tugadak Island, and we're currently diving at a water depth of 700 meters, and this is part of a series of water column transects that allow us to gather information about the water column, including the salinity, the temperature, and some of the biodiversity that's within the water column at these different levels. And so we should be finishing up the 700-meter water column transect shortly.
Yeah, and this in front of us here is a Poralia jellyfish. Nice bright red color. Red is pretty common in deep waters because red light is actually attenuated or bounced out of the water column pretty early on. So if you're a red color at depth, you're actually pretty much invisible. So it's a really good color for an organism to be. So we see a lot of red jellies, and when we get down to the benthic dives, we'll see a lot of animals that are red or some variation on red. This is a beautiful shot of this Poralia. We've seen quite a few of these today. Some even with chunks taken out for where something has been feeding on them in the water column. See that line of fine tentacles all around the outside of the bell. Up at these shallower depths, the water current is moving quite a bit faster than it was down at the deeper depths. So you might notice the water going past us quite a bit faster. It's a little harder to keep steady at these depths. You can see all those particles moving past us. Oh, a little fish. So that was the end of the transect at 700 meters water depth. So we'll be moving up to 500 meters water depth and then eventually 300. Sorry about that, I was mistaken. We have a few more minutes here of this transect. Little fish sitting in the water column here with its head up. I'm not quite sure what species of fish this is. I don't know if anyone in the chat room knows. Oh, so this is another one of those siphonophores, the Marius, the fire belt siphonophore. And looks like this one's had a little accident. Something's come into contact with something that's broken the pieces apart. These siphonophores are really interesting because they're actually colonies. So it's lots of different organisms all connected together. And siphonophores are pretty unique in that each of those little pieces is doing a different job. So some of them are for feeding, some of them are for moving, some of them are for reproduction. You can see this colony has just been broken apart. It's not quite as beautiful as the ones we've been seeing during this dive. Oh, look at all those little zooids that are broken up. This is the fire belt jelly. Fire belt jelly, the siphonophore. Okay, thank you, Nav. So that is the end of the 700-meter transect now, end of this transect. So now we'll be moving up into 500 meters of water depth, where we will do another transect in this mid-water dive.
Hi everyone, thanks for tuning in to our dive this afternoon, and I wanted to just give you a overview of the expedition and the dive that we're doing today. This is the third expedition of the Seascape Alaska campaign, exploring offshore waters of the Aleutian Islands area. We are currently located about 60.5 miles offshore from Tugadak Island, and we're diving at the edge of the continental shelf, which is a really interesting area. Our dive today started out around 2,200 meters, and we completed a series of water column transects where we could collect water samples and also survey some of the organisms that are occupying those different water depths. We are currently on our way to the next transect, which will happen at 500 meters, and we should be arriving at 500 meters in just a few minutes, at which point the ROV will settle out and we will take a water sample and then begin our transect. So again, thanks everybody for joining us, and thankfully we have some water column experts on the line with us that have been helping to identify some of the animals. Special thanks to Mike Vecchione for commenting on the line about many of these animals. I know we've all learned a lot from listening to him, and thanks again to everybody who called in to the chat and is providing some of their expertise. So we should be arriving at our transect shortly.
Oh, this looks like it might be a little glass squid. Yeah, George Matsumoto on the chat is agreeing. Oh, beautiful. And I believe this is what's called a glass squid. They look almost like a little glass ornament.
That's in the family Cranchidae.
Thanks.
Sorry, Rhiann.
No, thanks, Elena, for chiming in.
Yeah, that's beautiful.
Can see the back fin moving. Oh, and the eyeball. Look at that.
Yeah.
It's starting to focus on us, maybe, or it's moving. Have you seen that before, Elena?
Yeah, this is what we would call Bellinella borealis. It is closely related to Teonius tavo that they get down off Monterey.
It's really beautiful. Is the red in the middle there? Is that the pen?
Yeah, that is, they call it the liver, but it's actually the digestive gland. So what you're looking at is the two little crescent-shaped things next to the reddish-pink thing. Those are the gills.
Okay.
And then the digestive gland is the single organ sitting in the middle. You can see the photophores on the eyes. That's a great shot. Thank you so much. So there, that's the ventral portion of the squid that we're looking at. So you can see the funnel right between the eyes. So we have this beautiful set of photophores right by the funnel, and then right underneath the eye, like the eyeball. Oh, did you just see the eye? Oh my gosh, the muscular portion that's covering the eye, the cornea, just opened a little bit.
Yes, yeah, it's been opening and closing on both eyes. It's really amazing.
Yeah. Yeah, wow, this is phenomenal. Wow, that's beautiful.
So there's little glands under the eyes. You said those are the
actually
photophores?
Yes, those are the photophores.
Nice.
This is a very common squid, but we don't typically see them in such good condition, so this is a treat.
It's amazing. It's really amazing. Who knew squid eyeballs would be so exciting?
Well, okay, I did.
It's good to know, Elena.
Sorry, I couldn't resist.
No.
Thank you for that. Amazing footage. That was awesome.
You know, it's one of the really neat things about using the ROV for these transects is we don't, usually we see a lot of these animals that come up in trolls and they're collected and just don't look the best. And now we get to have a look at them in situ and see some of those behaviors. It's really amazing.
It is. Yeah, I love this.
A glass squid, yeah. So that one was a glass squid. Sitting on the back row here, we're still kind of reeling in the footage of that glass squid there, how good that was. This is another Paralia jelly. We've seen quite a few of these during this dive today, a few of them with bites taken out of the side of them as well. This beautiful red color. So the fine tentacles just as it pulsed there. lot of animals in the deep ocean are red. You might have noticed on this transect we've seen a number of organisms that are red, and that's a pretty common strategy in the deep sea to hide from predators. Red light is attenuated or bounced out of the ocean pretty shallow, and so there's no red light at depth, and so if you're this red color, you're effectively invisible to other organisms, so it's a great way to hide in the water column. The other strategy that's really common in the water column, a little bit more than on the benthos, is to be clear as well. It's quite energetically expensive to make your body clear. You've got to hide all your organs, hide all your bits and pieces. If you're red, you can kind of make things different shades of red. This Paralia is being bounced around by the current generated by D2. I have a feeling Mike Vecchione is going to be upset he missed that footage of the squid, and he comes back on.
Agreed.
So if you're just joining us today, we're on the first dive of the Seascape Alaska 3 expedition, and we're diving off of the Aleutian chain during this expedition in deep waters. Today today we're about 60.5 miles off the nearest landmass, and we started our dive today in 2,260 meters water depth, and we've been doing a mid-water dive today. So we started our first transect at 50 meters off the bottom in the benthic boundary layer. We then did a transect at 900 and at 700, and now we're in the middle of our transect at 500 meters water depth. And so what the pilots are doing with the ROV is we take the ROV up to a depth, and then they move forward at a steady pace, and we take videos and sometimes samples of organisms that we encounter in the water column. We've seen some really cool things today. We just saw a really neat glass squid. Seen lots of a couple of different kinds of jellies and ctenophores as well. So it's been a pretty exciting dive for our first dive of this expedition. This one is a collection target, pilots. This Tina 4 up top.
Hey, Okeanos, Mike back.
We hear you, Mike. Thanks so much.
Yeah, sorry, but I'm back.
We found a Tina 4 that might be a good sample target here, so we're getting ready and hoping that we might be able to collect it. Thanks.
Yeah, I see. I did see the Cranchiad, and I'm glad that Elena got on to tell you what it was.
We got it. We can see on the images here that we did, in fact, collect that sample.
That was a ctenophore called Bolenopsis, and it's been incredibly common on this transect, particularly in the deeper ones. So good to get a sample and really confirm what that is. Well, he's definitely squishy. I'm not sure he could be yours. This is another little jelly. I think it's another dinner plate jelly, but I'm just waiting for the chat room here to update for me. Bathychorus. Bathychorus. Sorry about that. Seen a lot of these ones here. Science doesn't need another image of that one. Yep. So it looks like another ctenophore, but maybe one that's slightly different. It seems to have really dense, long tentacles trailing out behind it. The ones we saw earlier are a little bit looser than that. Beautiful. So these ctenophores are comb jellies, and you can see those combs kind of iridescing along its body there, waving backwards and forwards, and that's what they use for locomotion, along with some propulsion often given from the inside. Look at those beautiful tentacles. So these trailing tentacles are for catching food. So they use them to capture food particles out of the water column. And they have stinging cells called cnidocysts on there. These cnidocysts look almost like little harpoons or barbs that shoot out as soon as prey goes by. It's beautiful.
They have sticky cells, rather.
Oh, I'm sorry, sticky cells. Yep, that's right.
But this, if you look at the body on this, you'll see there's two horn-like things that come out from the sides of it, and that's why it's called ctenoceros.
That's right at the head end there.
Like rhinoceros.
Yeah.
Yeah.
Yeah, of course,
George identified it as ctenoceros.
Yeah.
Orange ctenoceros, Google says.
Ctenoceros. Beautiful.
I think it's an undescribed species.
Yeah, of course, it's the jellies that have those harpoons, and the ctenophores have sticky cells. Yeah, this is a lobed or lobate ctenophore, it looks like. Oh, swim away. Going faster. It's almost like it's missing one of its lobes on the back end. Another part of this feast that's been happening down here in the deep sea.
I was just thinking the same thing.
Yeah. Thank you, Nev. Thank you, Nev. It's in the top center there. Going straight by us, yeah, that's okay. Might be a mucus string of some kind. Oh, yeah. Another siphonophore, potentially? Yeah, I think it is. I think the bell is down on the right-hand side on the bottom there. Yep. There it is. Beautiful. So this is a siphonophore, which is a colony of different zooids. And each one of these little animals that are together in this colony have a different job. So some are this bell at the end, which can be for locomotion or flotation. Then it will have other pieces that are for reproduction, and then other pieces for feeding.
Dougal's asking if it could be slurped.
Yeah, could we take a sample of this, or are we too far? Okay. The bell would be the important part, yep. Guess 4. Confirmed, I see some of it there. Yep. Okay. Thank you, pilots. Little fish here, another myctophid maybe? It's got a very dark head end there.
I think that's an owlfish, a bathy-lagged.
An owlfish, did you say? That was beautiful.
Yeah. They're jelly eaters.
Maybe the predator we've been looking for.
Their mouths aren't very big, so I don't think they've been taking the big chomps out of the jellyfish.
Okay. Maybe it's not the predator we've been looking for. It's a beautiful black head with those bright eyes.
Yeah. They're neat fish. When we were diving on the Mid-Atlantic Ridge, they were the most abundant deep fish that we found.
Interesting. So we're in about
sampling.
Yeah, we're about 500 meters of water here. Is this deep for their range, or kind of in the middle of their range?
No, I think it's pretty normal depth for them.
Okay.
Get deeper than that. I think the ones we saw at the Mid-Atlantic Ridge were around 900 meters, but I'm just pulling that out of thin air. I don't remember.
Beautiful little jelly here with four long tentacles. Maybe a Bathychorus from Dougal Lindsay. So if you just joined us here today, we're on dive number 1 of the Seascape Alaska 3 expedition. We're diving just off the Aleutian chain, very close to the Aleutian Trench. Today we've been doing a mid-water dive. We started down in the deep, 2,160 meters, about 50 meters off the bottom. And then we did a transect at 900, a transect at 700, and we're just coming towards the end of our transect at 500 meters. There'll be one more transect to go after this one at 300 meters before we bring up the vehicles and move on to our next dive site ready for tomorrow. Another siphonophore here, these colonial organisms that live in the midwater. It's just a little tiny one. Is that another little fish off to the left there? Another fish. What are you? Oh, it's another little siphonophore. Just zipping along. Very fast siphonophore. Thank you so much, pilot. So that's the end of our 500-meter transect. So now what we're going to do is move the vehicle up to the 300 meters, and we'll start another transect there. And that will be the final transect of the day in the midwater.
Hello, everyone. If you're just joining, we are on our first dive of Seascape Alaska Expedition number 3. We're currently located offshore of the Aleutian Islands, about 60.5 miles off of the island of Tugadak. And we started today out at a depth of about 2,200 meters. And we've collected a series of transects through different water depths in this midwater dive today. Starting at the bottom, we've took a transect at the 50 meters off the seafloor, and then we took another transect at 900 meters, and then another at 700 meters. And we just completed the transect at 500 meters, and we're on our way to our final transect, which will be conducted at 300 meter water depth. At each of these levels, we take a sample of the water. The samples of the water include analyses, or available for analyses for eDNA, and also we've taken numerous samples of various organisms that we've encountered along the way. And we should be reaching our next transect location momentarily. Thanks.
Thank you, Nav. So this is the beginning of the 300-meter depth transect. So if you're just joining us today, we're on dive 1 of the Seascape Alaska 3 expedition, and we're doing mid-water transects today, and we're on the last transect of the day at 300 meters depth. Looks like we're seeing another lobed ctenophore. Maybe lobed. Oh, maybe not. Dougal Lindsay from the science chat room is saying this is another Balanopsis, which we've seen quite a few of today. Oh yeah, what's that one? A little jelly. Fish. Oh, just gone out of sight, unfortunately. Oh no, what's that there? Is that a little squid or octopus? This is where I need Mike or Elena if you're online. Looks like another one of these little glass squid with his tentacles out. Oh, unfortunately just going out of our range there. Squid. Mike Vecchione says that, confirms that that's a little squid. two more of these low bait ctenophores. These ctenophores don't have those big, long, trailing tentacles like we've seen in some of the other species we've been looking at today. Instead, they have these two kind of fins that almost do like a frog kick to swim through the water. Ctenophores have certainly been quite abundant during this dive today at the various different depths that we've been working at. Yeah, it looks like another ctenophore. That one might have had a meal. It's got kind of a little bulb in the middle there where the stomach would be. Or maybe not. Hard to tell. These little iridescent lines that you see down the ctenophore, these are called the teens or comb rows. And they're made up of lots of little, almost like flappers that help it swim. You have confirmation of Dougal. Yes, that little part in the middle is where it's eaten something, had a meal, and is busy digesting on the inside there. These ctenophores just have one opening, so the food goes in, it's digested, and then whatever isn't digested or isn't wanted is spat back out again. Yeah, definitely seeing quite a few more of these ctenophores in this layer here. When we look in the midwater, we look in these different layers because each layer might be slightly different. You know, the water mass, even in the deep sea, is not one big homogeneous area. There's lots of different currents, lots of different flows that go through and creates kind of the stratification of the water. And each one of these water masses might be slightly different temperatures, different oxygen, different nutrient contents, and as a consequence, attracts different animals to live in these different layers of the ocean. Oh, a correction from Dougal Lindsay that these ctenophores actually do have a pore that they excrete from. They don't spit out the waste. They actually have an anal pore that the waste comes out of. Whereas the jellyfish, the Cnidaria, only have that one opening. Sorry about that. And looking at all these particles that we're seeing in the water column here, I feel like there are maybe even less particles at this depth than we saw down in the deeper parts as well on the last couple of transects that we've been doing. Still a lot of these particles, which are known collectively as marine snow. And marine snow is really what feeds the deep ocean. So in the deep sea where animals live far below sunlight, they're not photosynthesizing to get their food. Instead, they're relying on all these particles floating down from above, and these particles are a big mix of different things. We got mucus houses like we saw earlier today, parts of decomposed larger organisms, phytoplankton, zooplankton that have died in the upper ocean and are floating down, even sediments, carapaces of crustaceans, as Mike Vecchione was telling us earlier. There's this huge mix of different food sources that rains down in the deep sea and is picked out on the way down as well. A lot of animals eat this on its way down into the deep ocean. There's a little arrow worm swimming off to the left. Yeah, it might be another one of these mucus houses. That'd be a good zoom if we can do that. Oh, maybe not. No, this is this is another ctenophore. Just a slightly different angle. You can kind of see its wings sticking out there. And more of those black markings that we've been observing on a lot of these. Yeah, another great siphonophore there. We've seen quite a diversity of different siphonophores during this dive. These siphonophores are these colonies, collections of different organisms. Oh, is it a salp?
That's a salp.
Oh, it is. Look at that. A salp with his tail. Look at that. I thought that was a bell on the end with all the zooids going. Beautiful.
And it's got holes at both ends.
Yeah.
And it's got muscle bands if you look closely.
So what is the tail for there? Like, what are the different parts of the salp?
That's budding also, like what we saw with the doliolus. Dougal says that's mimicry of a siphonophore. So it's trying to fool something into thinking that it's a siphonophore.
Interesting. Yeah.
Which seemed to work.
Yeah, mimicry in the mid-water. And asexual reproduction, Dougal Lindsay is saying as well, that these could be asexually produced.
Budding like the doliolus did.
Yeah. Interesting.
A lot of things that try to look like siphonophores because siphonophores have very powerful stingers. And if you can mimic one in the area where vision is important, then you're less likely to get chomped on by a predator.
Nice. Yeah. Thank you, Nav. So we have about 10 minutes or so left of this transect here. We're in 300 meters of water. And if you've just joined us, this is the first dive of the Seascape Alaska 3 expedition. We are diving off of the Aleutian chain for this expedition. For this particular dive, we're looking at the midwater. We're actually on the last transect of the day. We're in 300 meters of water. And we've looked at 500, 700, 900, and then 2,160 meters of water depth. For each of these transects, we have the ROV drive along in a line for a fixed amount of time, and we look at all the midwater animals that we can as we're going. And we've been collecting some samples. And we've seen some really amazing things today. Lots of ctenophores. We had a bit of a ctenophore disco at one of our depths. We've got some amazing footage of a couple of different kinds of squid. We've seen some salps, and then lots of little jellies as well. So this has been a really great midwater dive, a good start to the expedition.
Dougal keeps noting down these tuscaroids. They look like a bundle of stuff hanging from some mucus. Those are radiolarians, so they're single-celled organisms, but they form a colony of a few that are all sort of bound together in that mucus, and they dangle below the other mucus.
So this is another one of those siphonophores with those big, long tentacles coming out the bottom. Beautiful. You can see the swimming bell there pulsating along. That's all those different individuals all pulsing, working together to get the colony out of our way. The giant D2 that's crept up on it.
Whether it could be collected. Can we collect that one?
Just waiting for confirmation. There it is. Yep.
You guys know that tonight when you handle the samples, you got to be really careful of the siphonophores.
That siphonophore was pretty fast.
Thank you, Navigator.
For those of you that might be joining for the first time today or just popping in, we are finishing up our first dive of Seascape, Alaska. We're currently located 60.5 miles offshore from Tugadak Island. We started our dive today at about 2,200 meters water depth, and we've slowly moved our way up to 300 meters water depth. We've completed a series of water column transects beginning down at about 50 meters off the seafloor. And we took additional transects at 900 meters, 700 meters, 500 meters, and we're just finishing up our 300-meter transect. At these transects, we take water samples, and we also take samples of some of the organisms that we encounter. And we've been very lucky to have members of our shoreside scientific community joining us today to narrate sections of the dive and share their expertise, and we really appreciate that. And we should be finishing up our final transect here shortly.
Yeah, I just wanted to say that we've just gone past several Burrowie ctenophores, gotten fairly good looks at them, and that's a different kind of ctenophore. It's not a sidipid, which is the kind with the two tentacles, and it's not a lobate. And Burrowie eat other ctenophores, so that they've got a mouth, fairly big mouth at one end, and they engulf other ctenophores and eat them.
Do
It's a different kind of ctenophore than what we've been seeing.
Do they eat jellies as well? Could it be our mystery predator?
I don't think they have the capability of biting. I think what they do is engulf their prey whole. But maybe Dougal can add to that commentary, since I don't actually know what I'm talking about.
That's okay. So we've actually come to the end of our transect today. So first off, I want to say a big thank you to all of our scientists on shore who have helped us through the dive today in this midwater dive. And thank you to all the ROV and video crew here on board, as well as the ship. I would like to say, as we proceed to explore the deep waters of the Aleutian Islands, we respectfully acknowledge that we are working within and around the traditional Alaskan native homelands of the Sukpak, Alutiiq, and Unongan. We acknowledge the native homelands and honor the indigenous knowledge and heritage of these coastal communities and their relationship with the ocean. We will work with care not to disturb the Alaska native uses of the ocean environment, and we look forward to building upon this indigenous knowledge to add to our collective understanding of the oceans. Thank you so much for joining us today from shore, and we look forward to a dive tomorrow. Tomorrow will be a benthic dive, and we're hoping to dive in a canyon area that has just been discovered using the multi-beam sonar. So thank you so much, and we will see you tomorrow.
And Dougal and George both added to the conversation about Burrowie eating other gelatinous stuff. And it turns out they can take bites out of things. They have giant cilia that act like teeth. But I don't know if that's what's been eating our Paralia and other big jellyfish. I'd like to add that I really appreciate everybody who's participated and contributed to this dive. It's been a great midwater dive. Thank you.
Yes, Mike, I would agree. Thank you so much for being on the line. You and Elena have been incredibly helpful in helping us on this exciting dive today. Thank you so much.